The names sound like they come from a story. Mitochondrial Eve, Y-Chromosomal Adam. They sound ancient and foundational, like the first two people in a grand epic. And in a way they are. But the story they tell is not one of a garden and a forbidden fruit. It's a story written in our DNA, uncovered by scientists, and it's about where every single one of us comes from. But let's get something straight right away. These two individuals were not Adam and Eve from the Bible. They were not the first man and woman. They didn't know each other. They almost certainly lived at different times, possibly tens of thousands of years apart. And they were absolutely not the only people on Earth when they were alive. They lived in a world populated by thousands of other humans, so if they weren't the first people and they weren't alone, who were they? The answer is simple, but what it means is profound. Mitochondrial Eve is the most recent woman from whom all living humans today descend, through an unbroken line of mothers. Y-chromosomal Adam is the most recent man from whom all living men today descend, through an unbroken line of fathers. They are not our only ancestors, far from it. They are just the two individuals whose specific genetic lines, the maternal and the paternal, have survived in everyone to this day. Everyone else's lines, for those specific pieces of DNA, eventually hit a dead end. To really understand this, we have to look at how we inherit our genes. It's not as straightforward as just getting a 50-50 mix from mom and dad. Some specific parts of our genetic code follow very strict separate paths, and those paths are what allow us to look back in time. Let's start with mitochondrial Eve. Inside almost every cell in your body, you have tiny structures called mitochondria. They are often called the powerhouses of the cell because they generate most of its energy. What's fascinating about them is that they have their own DNA, completely separate from the main DNA coiled up in the cell's nucleus. This is your mitochondrial DNA, or mtDNA. The inheritance of this mtDNA is unique. When an egg is fertilized by a sperm, the resulting embryo gets all its mitochondria, and therefore all its mtDNA, from the egg cell. The father's sperm has mitochondria to power its journey, but they are typically located in its tail, which falls off during fertilization. Even if a few get inside the egg, they are actively destroyed. The result is that you, me, and every other person on the planet inherited our mitochondrial DNA exclusively from our mothers. She got it from her mother, who got it from her mother, and so on, in a direct, unbroken maternal line stretching back through time. It's like a genetic surname that only women can pass on to their children, both sons and daughters. This provides a clean, traceable line, but there's another piece to the puzzle. The DNA mutates. Over generations, small, random changes occur in the genetic code. Most of these changes are harmless and just get passed on. Scientists realize that these mutations happen at a roughly predictable rate. This means mtDNA acts like a molecular clock. By comparing the mtDNA of two people, you can count the number of mutational differences between them. The more differences there are, the more time has passed since they shared a common maternal ancestor. In the 1980s, a group of researchers led by Alan Wilson and Rebecca Cann decided to use this clock to build a human family tree. They collected mtDNA samples from people all over the world representing a wide range of geographic and ethnic populations. They sequenced the mtDNA from each person and started comparing them. They built a tree, connecting the most similar sequences on small branches and connecting those branches to larger, older ones. They kept working their way back, finding the common ancestors for larger and larger groups of people. Eventually, all the branches of their human family tree merged into a single root. Every single mtDNA sequence they studied, from every person on every continent, ultimately traced back to one ancestral sequence. The woman who carried that sequence was named Mitochondrial Eve. Using the molecular clock, by estimating the rate of mutations, they calculated when she might have lived. The initial estimate, which has since been debated and refined but remains in the same ballpark, was around 200,000 years ago. And where did she live? The data gave a clear answer. The oldest and most diverse branches of the mtDNA tree, the ones with the most mutations separating them, were found exclusively among modern African populations. This meant that she must have lived in Africa. Everyone outside of Africa belonged to newer, less diverse branches, suggesting their ancestors had migrated out of Africa much more recently. This was powerful genetic evidence for the out-of-Africa theory of human origins, the idea that all modern humans originated in Africa and then spread across the globe. But this is where the biggest misconceptions arise. It's easy to picture Eve as the only woman, but that's not right. The key is to think about lineages dying out. Imagine a small village of our ancient ancestors. Let's say there are 100 women. In the next generation, some of those women might not have any children. Their mtDNA lines stop right there. Some women might have only sons. Their sons will have their mtDNA, but they can't pass it on. So those empty DNA lines also stop. Only the women who have at least one daughter will see their empty DNA line continue. Now, play that forward over thousands of generations, it becomes a game of chance. By pure random luck, over a vast amount of time, more and more of these maternal lines will eventually hit a dead end. A family line has only sons. A catastrophic event wipes out a family. A woman has no children. It's a process of pruning the human family tree. Eventually, if you go back far enough, you'll find that only one of those original 100 maternal lines has survived in the present day. Mitochondrial Eve is simply the name we give to the woman who was at the beginning of that one lucky surviving line. She was a normal woman in a population of thousands. She had no idea her DNA would be so successful. She was just one of many, but by chance, her maternal lineage is the only one that never ever broke. The story of Y-chromosomal Adam is very similar, just on the other side of the family tree. This story is written on the Y chromosome, the piece of DNA that, in humans, typically determines male sex. A person with two X chromosomes, XX, is female, while a person with one X and one YXY is male. The Y chromosome has its own special way of being inherited. It is passed directly from father to son. A man's Y chromosome is a copy of his father's, who got it from his father, and so on. It's a direct paternal line. Much like empty DNA for the maternal line, it provides a clean, traceable path back through history. Most of our other chromosomes are a jumbled mix of our parents' DNA. They swap sections in a process called recombination. But the vast majority of the Y chromosome doesn't do this. It's passed down almost whole from father to son, making it an excellent record of paternal ancestry. And just like empty DNA, the Y chromosome also accumulates mutations over time. It has its own molecular clock. So, scientists did the same thing they did for Eve. They collected Y chromosome samples from men across the globe. They compared the sequences, counted the mutations, and built a family tree of paternal lineages. And just like before, they found that all the branches eventually converged at a single root. This root belonged to the man we call Y-chromosomal Adam. He is the most recent common ancestor of all living men through the paternal line. He, too, lived in Africa. The data from his family tree also pointed to an African origin for all modern humans, reinforcing the findings from the mitochondrial Eve studies. This provided two independent lines of genetic evidence that told the same powerful story of our beginnings. However, for a long time, there was a confusing wrinkle in the story. Early estimates of when Y-chromosomal Adam lived were much more recent than for mitochondrial Eve. Some studies placed him as recently as 60,000 years ago, while Eve was still estimated at around 200,000 years ago. This created a paradox. How could our most recent common paternal ancestor have lived so much later than our most recent common maternal ancestor? It didn't make sense. It led some people to question the whole idea, but this is how science works. It's a process of refinement. Later studies using more sophisticated techniques, more samples, and a better understanding of mutation rates in the Y chromosome revised the timeline. It turns out the Y chromosome might mutate at a different rate than initially thought. Current estimates now place Y-chromosomal Adam somewhere between 200,000 and 300,000 years ago. Puts him in a much more compatible time frame with mitochondrial Eve. While they were not contemporaries who met and had children, they likely lived in the same general era of human history. The same misconceptions that apply to Eve also apply to Adam. He was not the first man. He was not the only man alive at his time. He was simply the lucky one whose paternal lineage, by chance, is the only one that has survived to the present day. Think about it again. A man might have no children. His Y chromosome line ends. He might have only daughters. His daughters carry his other genes, but not his Y chromosome. So, his paternal line ends. Over countless generations, all but one of these Y chromosome lines eventually came to a halt. Y-chromosomal Adam is the man at the top of that single, unbroken chain of fathers and sons. So what does all of this actually tell us? Why do these two ancient individuals matter so much? Their existence as scientific concepts provides a powerful framework for understanding the story of humanity. They are the twin pillars of the out of Africa theory. The fact that both our ultimate maternal and paternal lineages trace back to Africa is overwhelming evidence that modern humanity was born there. Furthermore, the branches of their family trees allow us to map human migration across the planet. As small groups of people left Africa and moved into Asia, Europe, and the Americas, they carried their specific mtDNA and Y chromosome markers with them. Over time, new mutations occurred in these new locations, creating new branches on the family tree. Scientists can track these genetic markers known as haplogroups and follow the paths our ancestors took. We can see the genetic trail of the first people to populate Australia, the waves of migration into Europe, and the incredible journey across the Bering Strait into the Americas. It's a detailed history of our species written in our own biology. These studies also tell us a profound truth about human diversity. The greatest amount of genetic diversity in our species is found within Africa. This makes perfect sense. Humans have lived in Africa for the longest time, so they've had more time to accumulate genetic variations. The small groups that left Africa to populate the rest of the world carried with them only a fraction of that original diversity. This means that two people from different villages in Africa can be more genetically different from each other than a person from Europe is from a person from Asia. It's a powerful scientific rebuke to outdated and unscientific notions of race. Genetically, we are an African species. There's one more subtle but important point to understand about Eve and Adam. Their titles are not fixed. Mitochondrial Eve is not a specific person locked in time, but a designation that applies to the most recent common maternal ancestor for the people who are currently alive. It's a floating reference point. Imagine if tomorrow a global catastrophe wiped out everyone in Asia and Europe. The new group of surviving humans, all of African descent, would have a different, more recent mitochondrial Eve. The old Eve would still be an ancestor, but the most recent one that connects everyone would have changed. In the same way, the mitochondrial Eve for humans living 50,000 years from now will likely be someone who lived much more recently than our Eve. As some lineages die out, the point of common ancestry for everyone who remains moves closer to the present. The same is true for Y-Chromosomal Adam. The titles are always relative to the current population. It's also crucial to remember that while these two lines are special because they are easy to trace, they represent only a tiny fraction of our total ancestry. You have 2 parents, 4 grandparents, 8 great-grandparents, and so on. Every generation you go back, the number of your ancestors doubles. If you go back just 30 generations, about 750 years, you have over 1 billion potential ancestor slots on your family tree. Many of those slots will be filled by the same people, but the number is still immense. Mitochondrial Eve and Y-Chromosomal Adam are just 2 of those billions of ancestors. We inherit genes from all of them. Every other gene in your body on your other 22 pairs of chromosomes has its own unique history and its own most recent common ancestor. Those ancestors all lived at different times and in different places. Our genome is not a single book with one origin story. It is a vast library of stories, each with its own beginning. In the end, mitochondrial Eve and Y-chromosomal Adam are not a couple. They are scientific concepts, anchor points in the deep history of our species. They were real people, a man and a woman who lived and died in Africa hundreds of thousands of years ago, unaware of the incredible genetic legacy they would leave. They were not special in their own time. They were just part of the human tapestry. But by the random lottery of inheritance and survival, their maternal and paternal lines, and only theirs, managed to weather every storm, every migration, every famine, and every generation to find a home in every single one of us today. They remind us that for all our superficial differences, every person on this planet shares a common thread, a deep and ancient connection that is written into our very cells. They are the start of a story that is still being written, a human story that belongs to us all.